better. And so I shared a little bit of my academic interest with Angelica, and I have worked a little bit on depression, but from the more from the treatment perspective, the mechanism of action of antidepressants. But also in our lab, we're working on some neurodevelopmental disorders such as autism spectrum disorder. So today I will share with you some aspects of the genetic basis of autism spectrum disorders, and perhaps we can discuss later a little bit of what we are specifically doing. But for those of you that are not aware, autism spectrum disorders is this umbrella term that was created when the latest edition of the Diagnostic and Statistical Manual for Psychiatric Disorders was published. The version 5 was published. Basically, they were trying to put together a number of disorders that had previously been separated in different categories, but now they were being there was this recognition that there was no real distinction between them, hard distinction between them. It was a continuous, it was a spectrum. And although definitions are perfect, and we can talk about that later, this was an attempt to sort of like update the definitions about autism spectrum disorders. But generally speaking, these are disorders that are diagnosed in children as early as when they're 2 years old or a bit later. The core phenotypic characteristics of these disorders is social communication or deficits or other aspects of social behavior or social interactions that are deficient in these children, and repetitive behaviors and interests. So these children display a lot of sort of stereotypies, and the degree of the intensity of these symptoms, it varies a lot. So some children actually can develop fairly at the level of a neurotypical individual. This is an individual without any psychiatric disorder. You require a lot of assistance for the daily living. So there is a wide variety, and heterogeneity is one of the key characteristics of psychiatric disorders. Like the definitions are so broad and so vague that it encompass people with very, very different manifestations of the disorders, although there is the notion that there is some underlying similarities. And as you can see from this diagram here, there is a lot of comorbidities with autism spectrum disorder. And again, we're talking about children here, not adults. I mean, obviously, these children grow up to become adults. So some of these comorbidities develop or change over time. But there is a lot of language disabilities. Seizures is another aspect that has been highlighted in several. There is a lot of irritability symptoms. There is mood disorders, sleep disorders, attention and hyperactivity. So ADHD diagnosis is highly comorbid with autism. But all individuals diagnosed with autism will have, in one way or another, these two core symptoms. There are no treatments for autism, particularly for the core symptoms. Currently, children are treated with some form of psychotherapy, and there are some repurposed treatments, for example, antipsychotics that are used to treat some of the comorbidities, never the core symptoms. So, and also we have to highlight some, many of these children also present gastrointestinal disorders, so a lot of problems with the digestive system, which is present there. So it might not be part of the core symptoms, but it's something that they suffer. So a little bit of more data. There's a worldwide prevalence estimated between 1% to 2% of the population. Not everywhere in the world there are accurate estimates. In the US and Canada, recent estimates put the numbers around 1 in 44 or 1 in 50 for each country respectively. In Mexico, there's no real instance data. There was a study published in 2016, that's the only one I could find, in León, Guanajuato, and they estimated a prevalence of 1 in 115, so a little bit lower than the rest of North America. There's some more or less reliable data in Europe and in East Asia, in particularly Japan and South Korea, but and Australia as well. But outside there's usually not very much reliable information. What we've noticed in most places where it's been studied is that there's strong sex bias. So males are 4 times more likely to be diagnosed than females. This is boys versus girls. And for example, in Canada, this means that the prevalence in boys is 1 in 32, which is much higher than the prevalence in girls, in 1 in 125. There are some biological explanations for this discrepancy that I will mention later. There's also the idea that for some cases, the diagnostic criteria may have been developed to recognize symptoms in males, but not in females. So there are some, sociocultural aspects that define our behaviors, and they might have an impact in the way that the diagnoses are made. But there are also some biological basis for this. So one thing that is very interesting interesting from a scientific perspective, but perhaps not for the population, is that the prevalence has increased like gradually over the last, since, for example, in these statistics from the CDC, from the United States, and from 1992 until 2010, sorry, from 2000 to 2018, you see that the prevalence goes from numbers 150 more or less, or down to the number that I just mentioned to you, 1 in 44. And this gradual increase, well, it's obviously puzzling, but one of the reasons for it might be increased awareness and changing diagnostic tools. So 20, 30 years ago, the appreciation and the diagnostics of autism much more different to what we have it today, and this had probably contributed to this increase in the rates of diagnosis. Although it's there's also the possibility that alongside with this there might be environmental factors that have changed or increase in magnitude over time, but there's no evidence for that at the moment. Anyway, going to more to what we are talking about today, the genetics of this. So as Maria mentioned, and you will hear this for most psychiatric disorders, genetic and environmental factors are important, and their interaction is important. That is true, and at the same time, it's become, it's true for everything. So it's become a little bit not that informative. So obviously what the research is trying to understand is what are those genetic factors, what are those environmental factors, and when they are interacting, when they're not. So epidemiology, it's crucial for defining this more in a way that it will become something that clinicians can use, etc. So autism is one of these examples that is sort of like on the extreme, because the genetic factors are widely recognized and is considered one of the psychiatric disorders with the highest contribution from genetics compared to other disorders. For example, perhaps Angelica will mention depression, or stories is slightly different. So heritability is one of these parameters that geneticists like to use, and it's basically it's a measure of how much of the variation that you observe out there in the population is explained by genetic factors. So the higher the number, it means that genetic contributions is much higher. And for autism, it's been estimated there. I mean, you see a range, but it's between 40 and 90%. So probably the real number is around 70 or 80%, which tells you like a huge percentage of the at least phenotypic characteristics of autism are defined by the genetics of the individual. And these, classical genetic studies with twins, where you see what's the percentage of individuals affected when they had a twin, an identical twin that was diagnosed with autism, and these numbers are pretty high for autism, 70 to 90%, and then the number decreases with the divergence in genetic similarities. So fraternal twins, it drops down to 30%, etc. So this is some of the highest numbers that you see for a number of psychiatric disorders. So early studies started identifying some of the genes associated with or linked or causing autism actually, by focusing in rare monogenetic disorders. So some around 3 to 5% of all autism cases can be attributed to one single genetic cause. But this is the exception, as you can see from the number. And these typical syndromes with high rates of autism include tuberous sclerosis, not to confound with multiple sclerosis. This is tuberous sclerosis. Rett syndrome, Fragile X syndrome, and other syndromes that have high rates of autism. But other characteristic of these mutations is they're syndromic. That means not only autism spectrum is present, but also these individuals have, for example, some physical abnormalities, etc., that accompany the autism diagnosis. So there are more complex disorders than what you will call an idiotypic autism case. Then with the when the next generation sequences start to be used, and then larger populations of individuals were started to be analyzed, and genome-wide association studies start to be implemented. This is sort of like a success story in psychiatry for these disorders, because they've been able to identify a number of genes that has associated. So just briefly, they can identify a lot of de novo single nucleotide variants and copy number variants, and I highlight de novo because that means they weren't present they are not present in the parents of the individuals that are affected. They appear only in the individual. So most likely they were mutations that were generated during gametogenesis. So when the sperm or the egg of the parents were being generated and meiosis was occurring, that's when these mutations appeared, and they will only passed on to that particular offspring, and not, they're not being inherited to all the offspring. So that happens quite a bit, and it contributes to approximately to 20 to 30% of the cases. There's other, autosomal recessive genes and mutations in regulatory and non-coding variants that are also contributing. But these de novo SNVs and CNVs are really what it's been identified as a large contributor to this autism. So there is a repository for all of these findings. It's called the SFARI it's a SFARI database. It's a charity that also gives a lot of funding for autism research in worldwide, but it's based in the United States. And they have made a huge contribution in the field by cataloging all of these genetic variants that have been identified. And they have curated them and then classified them according to how much evidence is out there for their involvement in autism. So they have put together a list of around 1,000 genes. It sounds a lot because, it's estimated that humans, we have 21,000 protein encoding genes, and about 1,000 of them can have mutation associated with ASD, but not all of them contribute the same way. So they've classified them in 4 categories: syndromic, category 1, category 2, category 3. 1, 2, and 3 are mutually exclusive. Syndromic can encompass some of the other categories. So basically, syndromic are these mutations that are causing autism, but are also causing other types of physical alterations. For example, typical example, Fragile X syndrome. And I'll finish quickly. I'm sorry if I'm going over time here. And then the category 2, it's just high confidence genes, so with a lot of genes with a lot of mutations. Genes that have been replicated in several genome-wide association studies. So the cutoff is more than 3 mutations, and the other 2 categories are 2 or 1 mutations respectively. And this is taken from their website, so around 200 genes are some of these high confidence genes that have been implicated. And one thing of note is a lot of these genes that are heavily implicated are in the X chromosome. Therefore, males are more affected than females because genetic males, like myself, have only 1 X chromosome, whereas females have 2. So that contributes to certain levels of resilience. And this is just broad scopes, just an idea of the genetics of this disorder. And I will stop now and give the floor to Angelica. Apologize if I went over time.
No, it's okay. Thank you very much, Dr. Aguilar. Very interesting, the data that you shared with us. And now we want to hear Dr. Angelica Torres Berrio. to see what she has to say. And later we're going to open questions from the students, and I have a couple of questions as well. So, Dr. Torres, yeah? Okay.
Yeah, I was muted, right?
Yes, you were muted, yeah.
Okay. So hi everyone, thank you very much, Angelica, for inviting me today and participating in this event, and everyone who is connected or part of the audience. Today, I would like to talk about depression, which is the main focus of my research. And can you see the entire presentation?
No, not yet.
Right now,
No.
Okay, perfect. So the main focus of my research is to understand the role of stress in increasing vulnerability to several psychiatric disorders, in particular depression. And depression is a highly heterogeneous disorder with an important genetic contribution. However, it interacts with environmental factors. So just as a brief introduction, depression is highly heterogeneous. It affects a significant percentage of our population, approximately 5 to 7% of the population worldwide. In this case, women are actually more susceptible to suffer from this disorder, actually twice more vulnerable to suffer from depression. This obviously leads to a significant cost in our health system every year because of either the days that are lost for inability to work, etc. The depression increases the risk for suicide, and unfortunately, there is a low response to the current antidepressant treatments. So it's very important to understand the neurobiology of this disorder. Again, as I mentioned before, it's highly heterogeneous. It involves alterations at the cognitive level, at the emotional level, at the motivational level, and still we haven't been able to develop antidepressants that can be useful for a significant number of patients. If we draw a line, we can see that early symptoms of depression can emerge in adolescence, sometimes earlier before. However, these symptoms can disappear spontaneously in most of individuals. However, in those who are highly vulnerable, these symptoms not only can persist throughout life, but also can become more severe, making it a very debilitating condition. If we want to understand the neurobiology of depression, it's important for us to pay attention to different brain regions. It's studies from postmortem brain tissue, from neuroimaging, animal models of stress and depression-like behaviors had identified different brain regions, in particular the prefrontal cortex, which is highly involved in cognitive function, planning, decision-making, the nucleus accumbens in combination with the ventral tegmental area, which are very important for motivation, the amygdala, which is important for emotion response, the hippocampus, which is involved in memory, and the HPA axis, which controls the response to stress. But what causes alterations in these brain regions is something that we are still investigating. So we know that depression, that different factors are involved in the neurobiology of depression. We know that genetic factors are involved as well as environmental factors. For example, family studies have shown that individuals that suffer from depression usually have other family members that suffer from this condition. Also, we know that environmental factors such as traumatic experiences are a high risk factor for developing this depression. However, not all individuals that are exposed to traumatic experiences develop depression. So epigenetic factors are actually a very interesting link that mediate between genetic and environmental factors. So different studies using twins, identical twins, or parental twins have shown that the genetic contribution for depression is approximately 37%, which is significantly low if we compare to other psychiatric disorders, as for example, in the case of, as Argel mentioned before, where like, for example, autism or schizophrenia, which have like genes that are like highly involved in these disorders. In the case of depression, it's just at 37% approximately. And new studies that involve genome-wide association studies actually try to identify different type of variants that can be associated with these conditions. So specifically, if we have 3 individuals that share a specific gene, they can show one specific variation in a single nucleotide. This is called polymorphism. The rest of the sequence of the gene is identical. However, this small variation can actually have implications in how the gene functions. And different studies using this type of GWAS samples can locate different type of polymorphisms that can be associated with depression. For example, this is one of the most cited studies regarding the genetics of depression, identify different genes that are highly associated with different traits associated with depression. For example, in this case was the perception of well-being and also neuroticism, for example. This is a very interesting study. However, one of the disadvantages of genetic studies is that, well, we have advantages, like, for example, they allow to discover new genes associated with depression and facilitate to identify these genes and see whether we can associate them with environmental factors. However, they don't necessarily involve causality. The fact that a gene is associated with depression doesn't mean that it causes depression. Actually, depression is so heterogeneous that the single contribution of a gene is minimal. It requires multiple genes to be disrupted, let's say, at the same time. So these studies, for example, don't include comorbidities and required a large number of samples. Actually, like we're talking about thousands of individuals, like hundreds of thousands of individuals to conduct these studies. And this makes it difficult, for example, in the case of Latin America, there are no studies of the genetics of depression in Latin America. Actually, I think there is a group in Mexico who's trying to develop this type of consortium and conduct this type of studies in psychiatric conditions in Mexico. But during my PhD at McGill University, I worked with our Mexican researcher, Dr. Cecilia Flores, and we were interested in a specific gene called DCC. DCC stands for deleted in colorectal cancer. This is a review in which we look at different studies looking at polymorphisms of this particular gene, DCC. We look at the number of individuals that were included in each of the studies. And one of the genes that highlighted the most, that were highly replicable, was DCC. Different polymorphisms and different regions of the gene were actually associated with depression. What is interesting is that DCC is a gene involved in neurodevelopment. During early development, DCC plays an important role in guiding where axons grow to specific targets. But later, it's involved in synaptic pruning and refinement of specific circuits. Later in adulthood, it's involved, for example, in synaptic plasticity. And during my PhD, I demonstrated that alterations in DCC in the prefrontal cortex of either postmortem brain tissue or animal models exposed to stress, they exhibit increased expression of DCC in the prefrontal cortex This was associated with disruption in the synaptic plasticity in the structure of pyramidal neurons in the prefrontal cortex, and this was regulated by a specific microRNA called miR-12. And in contrast, when there were optimal levels of DCC and optimal levels of these controlling microRNA, we actually were able to demonstrate that this promotes resilience to depression and also to stress. So in this perspective, it's important to have a multidisciplinary approach to understand the neurobiology of complex disorders such as depression. It's not only important to look at what happens at the genetic level with genetic studies, but also we need to go to the bench and sometimes use animal models to try to see whether what we can find in genetic studies actually have a functional role in the development of this disorder. These type of approaches actually facilitate not only the communication between researchers, but also building of new lines of research, and hopefully this could lead to the development of new antidepressant treatments. And with that, I would like to thank you, Angelica, for inviting me to talk today. And if someone has specific questions, this is my institutional email, and then this is my Twitter account. If I usually just retweet information regarding grants and organizations that support underrepresented minorities in science. Thank you very much.
Thank you very much, both of you. As I mentioned at the beginning of this roundtable, we are so excited to have you here because we know that you work in a very excellent way in your specific interest of research. I don't know if in the audience you have questions for starting this discussion. The idea of this roundtable is that we can ask the experts things that we maybe misunderstand before about genetic components in mental disorders. Or I don't know if you want me to start with certain questions that I have. Dr. Kathy, is there a question over there?
Is there any question, guys? Or we want Professor Acosta to start the discussion? Just to let you know, there was the queue up to the 101. I need to send them home. So just to let you know.
Thank you, guys. You're so popular. Oh my goodness, rock stars here.
Yeah, Professor Acosta, please, you can start, and then I'm gonna try to facilitate here the part.
Thank you, Dr. Kathy. Here, there is a question in the Zoom session from Patricia. Patricia, could you please unmute yourself?
Hi, is the Gino of depression dominant or recessive? That's my question. Nowadays.
Thank you, Patricia.
No, in this case, in this type of studies, we can see, we cannot really tell if it's dominant or recessive in the context of depression, but we can tell where specifically this polymorphism is occurring. For example, whether the polymorphism is occurring at a specific gene, a specific location of the gene, what is involved in, since this is a receptor, for example, if it's more in the transmembrane domain or like a intracellular domain, extracellular domain. But in this case, we cannot tell whether in this type of studies, we cannot see if it's specifically recessive or dominant.
Good question. Thank you, Patricia. Any more questions here in the Zoom session? So I have a couple of questions that I want you both to answer if it's possible. Yes.
There are 2 questions from the Zoom.
Oh, okay. Yeah, I don't see it. Okay, is Dan and Mario. Okay, who wants to start? Dan, do you want to make the question?
Yes, I just have a quick question. I did last semester a research about this topic, and I found that, for example, if a patient has a mental disorder such as depression or the other mentioned, like their children can develop not only the same but others types of mental disorders. Is this true? And like, how possible is this to happen? Like that, I was curious to know.
Who wants to
Couldn't hear very well that you're asking if there you can be diagnosed with more than one disorder. Is that the question?
No, for example, if someone has a mental disorder, yeah, for example, is this area, I found that is hereditary. And first, if this is true, and second, like, how possible is like to my children, like to, well, not mine, but an example, to develop the same as the patient or other mental disorders, like about these genes and all this, like make me wonder about that.
Angelica, you want to start or?
I mean, I can talk about depression and anxiety. And for example, in these type of disorders, we know that like depression and anxiety, they run within the family. However, not all individuals suffer from this condition. That's why it's highly heterogeneous, and that's why the genetic contribution, it's much smaller in these disorders as compared to other psychiatric disorders. For example, schizophrenia, where like the genetic contribution is much larger. Argel can talk about more like about those disorders. But for example, in the case of depression, we know that environmental factors play a very, very important role in the development. So it's like the combination of both. But yeah, there is a genetic component, of course.
Yeah, if I can add something, yes, as Angelica said, there's where diagnoses are very, very frequently, and actually they've been used for genetic studies a lot. But as Angelica also mentioned, often the diagnosis you can see the pedigrees of the family in these reports. And for example, you will see bipolar disorder, bipolar depression, schizophrenia, bipolar disorder with some psychotic type of symptoms. So, and all of those diagnoses are there. So there is something in genetic in the family, but it doesn't define one particular disorder. The problem is that disorders are also, the definition of each disorder is not hard. So you have some overlap, some continuum, for example, between depression and bipolar disorder. There is a lot of overlap, and sometimes it's difficult. And sometimes somebody's diagnosis will change over time, simply because for bipolar they didn't have the manic symptoms at some point when they were diagnosed, so they went, okay, it looks like a depression. Then when they express these symptoms, okay, the diagnosis changes. And sometimes it reflects simply how the disease is evolving over time. In the case of autism, what they have found in many cases is that a lot of the mutations, a great percentage of the mutations are de novo. That means they came from the parents, but the parents didn't have the mutation. They came from the germ, the sperm or the egg of the parents. So the mutation happened after the parents fully developed without any health issues, and then because of mutations during the process of meiosis, of production of the germ cells, then you can have an affected offspring and affected children. But that's another extreme. Yeah.
Okay, thank you very much. Ulises, you have a question as well.
Yes, Professor. My question is related to genetic studies. You
previously mentioned that one of the limitations or some of the limitations causes very few studies with Latin American populations. And my question is, what difference in results are we expecting in contrast with other populations in the world?
I would say that is a $1 million question. I think it's very important to do these type of studies in Latin America to see whether a specific, like as Argel was mentioning before, like the pedigrees, may differ, like, like the environmental conditions in Latin America are completely different as what happens in North America, that would make a specific genes to be probably more expressed or not, depends. But in terms of genes, at least in the context of depression, I mean, I would expect genes that are like highly common in overall human studies, like the serotoninergic receptors could be altered in Latin American population as well. But GWAS studies are important in Latin America to identify novel genes that haven't been already described in populations, in Caucasian populations, pretty much, because most of these GWAS studies are actually done in Caucasian population, like white people that are recruited for these type of studies, which is completely different. I mean, it's different from what Latino people are. I don't know if Argel wants to add on that, but yeah, that's definitely a way to highlight the importance of conducting these type of studies in our own population.
I completely agree with what Angelica mentioned, and I will add a couple of things. That one, it is clear that most cases or most psychiatric conditions are polygenic. That means you have a small contribution of many genes. To be able to detect that, as Angelica mentioned, you need very large populations in order to be able to pick up sort of like the signal out of the noise. If you to have a decent representation of humankind, ideally you will kind of try to encompass all the variation that we have. Like human population is quite variable compared to, well, if I compare to my lab mice that basically are twins from each other, we are extremely variable genetically speaking. So when you focus your studies, as Angelica mentioned, in white people from North America or Western Europe, you are effectively making a huge statistical error that is subsampling. You are drawing conclusions and making assumptions about biology by subsampling. So from that perspective alone, it just like, the field is screaming for diversity, and it's not, it's not only some left-leaning liberal position. It's simply from a statistical point of view, these studies, what they've been effectively doing is, just taking a subsample of the entire population and then assuming that this is the standard, and that couldn't be far from true. Like you need to basically look and try to do. And this is this is a very important consideration, and it's there's a lot of talk about it, particularly in the States, even in Canada, perhaps in some parts of Europe, about it including more diverse populations. The typical gap is Latin America and Africa are places where there is no sampling. Some parts of Asia, but Asia is developing much faster than Africa and Latin America in terms of scientific studies. So they are doing their own thing as well. But we have these gaps here with hundreds of millions of people that are not being considered, and then we don't know if the findings that we're going to apply for everybody, or it's just false positives we subsample, so positive problem subsample, sample like or you skew this to a certain populations, then whatever you observe is like, well, it's just very specific to one small population, but not relevant for many other people.
Yeah, and I think this is also important in terms of antidepressant treatments, for example, because there are some studies showing that specific polymorphisms can be associated with high response to antidepressants. But for example, if for some reason in Latin America is more prevalent certain type of polymorphism in which those antidepressants are not effective. So, but we don't know. So that's why it's very important to conduct these studies. We basically don't know. I think that there is a study published a couple of years ago. It was in Peruvian population, and it was not in depression. It was more like post-traumatic stress disorder, PTSD.
Okay, very interesting. Regarding antidepressants, there's another question here in Zoom. So Sebastian, please, I will leave myself for the end.
I just had a question regarding antidepressants, because I was shocked that at the stat Dr. Torres gave. So why do antidepressants have such a low recession rate? Why are they kind of not as efficient as we would expect?
It's my course, and you will know it.
So there are different explanations. Some of them, they can be the neurobiology of their action. For example, antidepressants usually take up to 2 weeks to have a specific effect. If you combine these with environmental factors, for example, an individual with depression going through a hard environmental stressful event that can also reduce the efficacy of antidepressants, probably individuals with a specific, as I mentioned before, a specific polymorphism in which antidepressants maybe not have it can be as effective as in other type of population. So yeah, there are different ways in which we can explain why current antidepressant treatments are not completely effective. We know, for example, that there's new efforts to give other options in patients that suffer from treatment resistance, meaning individuals that go to a specific course of antidepressants, then go to another course of antidepressants, and then finally they go to a different treatment. In this case is ketamine, which is a topic in which Argel is an expert. But ketamine, for example, has antidepressant effects, but it's not the first option that psychiatrists take when an individual goes to the clinic. So yeah, so definitely there's a lot of research to continue doing to try to uncover this. But one of the answers is that yeah, antidepressants usually take so long to have an action on individuals that some individuals, they just in some contexts, some people just drop the use of this treatment, the side effects as well.
Yes, just to add a little bit, what we try to mention here is how heterogeneous the disorder is, and this is not a trivial thing. This is not, oh, well, different, great. It also impacts treatment because it's probably what you are defining as depression. Well, first of all, there is no biological correlate of depression. Like, it's all based on self-reports. If somebody's feeling sad, and they will tell you, oh, I've been feeling terrible for the past 5 weeks. Okay, so you go to a psychiatrist or a psychologist, they run a series of questionnaires and they will diagnose you. But there is no if you have a liver disease or a kidney disease, there are some markers in the blood that you can follow for diagnosis and for treatment. Nothing like that exists for any psychiatric disorder. So that limits obviously what you cannot predict whether an individual will respond to a treatment or not. So it's a trial and error thing. So you give them, that's what Angelica mentioned, these rounds of treatments. You give them a first round, you have to wait around 6 months to see if it works or not. It's a long time for somebody that might be feeling depressed. for example, for depression. And then it doesn't work. Okay, let's try this other one. And again, the same thing, 6 months. And then when nothing has worked, okay, there are other alternatives, but obviously we have to first try. But again, there is no way to predict. And a good number, somewhere between 50 to 60% of people will respond to the first line of treatment, the SSRIs, the SNRIs, etc. But the other, 40 to 30 to 40% won't. And then, for example, now there's ketamine, at least it's approved in Canada and the States and other countries. But even for ketamine, it's not, I think there is a percentage of people that don't respond. So we don't fully understand the biological basis, therefore we cannot predict if something will work because we don't know if we're targeting the right thing or not. So it's a trial and error thing.
Okay. Thank you, Dr. Aguilar. Valles, you have a question. Please go ahead.
Yes, hello. Good afternoon to everyone. I have a question related to autism. I know that there are a lot of geniuses that have autism, like for example, Elon Musk or Darwin was considered to have autism also. Is there any reason for this? Maybe like a genetic factor?
So you mean some very smart people also have autism? Is that what you're referring to?
Yes, like a lot of geniuses
Yes.
have been
Oh,
have
gotcha.
autism.
I mean, probably just simply because now we know that autism can be close to 80 to 90% genetic variation. So probably that doesn't mean it was inherited, as I just mentioned. We can acquire, mutations can be acquired during meiosis after their parents were fully formed and mature adults. Then they can still have a child with autism because of mutations that happen in the germline only. Now there is a certain even with schizophrenia, because we know of a couple of cases of very smart people that they're geniuses, if you want, or extremely smart in their whatever, in scientific fields, and then they've been diagnosed with some disorder. And there's the idea of the autistic savant, these individuals with incredible mathematics abilities, yet they have autism. There is certain correlation, but unfortunately, for example, for schizophrenia, the most common thing that you find with schizophrenia is lower IQ. So those extraordinary individuals that have both, they're extraordinary also because they have both. How did they achieve that, or how did they develop into that? It's certainly something that we don't understand yet. But and a bit with autism, in the case of autism, you do have certain population that they may have these social impairments and their repetitive interest, but they do have an extraordinary ability with mathematics. But it's not something that you can generalize with to all people of autism. So a large population of people with autism actually has the opposite, has very low IQ, cannot function properly, cannot communicate, cannot verbalize. So they're children that can't learn to speak, etc. So you have both. So the fact that we see, these extraordinary examples, they are extraordinary, but at the same time, that just means they're not very common. So a lot of these individuals with some of these psychiatric disorders actually will suffer of the opposite. So if you look at an averages, which is something that we tend to do a lot in research, for better or for worse, the average IQ of many of these patients is actually lower than neurotypical population. Again, that doesn't mean there are exceptions, and that doesn't mean there are individuals with extraordinary ability, mathematic abilities, or this sort. So all of these complicated answer is just to say that it's not the norm. It's not the what you usually see in your average autistic patient with autism. But there are some extraordinary cases.
Thank
Okay,
you.
thank you.
Thank you, Valles, for your question. Yes.
There's a question here.
Okay, please.
Can you hear me or should I go downstairs?
I think a little bit closer, right? Because we cannot hear you properly. Barely, we can barely hear you.
The microphone is over there.
So you mentioned there was a higher probability that females had depression, but I wanted to know, is there a biological basis or could this be attributed to gender biases interfering with the diagnostication?
Well, this is right now the main focus of research. I don't know how it's in Canada, but here in the United States, it's no longer an excuse not to include females and women in the studies. Usually, for many years, female were not included in studies because of hormonal variation, etc. But psychiatric disorders such as depression, for example, affects women. In the lab where I'm working, for example, there is a postdoc who studies specifically sex differences, and she identified long non-coding RNAs that are associated with specifically female depression. But this is just a way to begin to uncover the neurobiology of this disorder, at least from a female perspective. But yeah, one of the limitations that for many years research have neglected is the use or like the inclusion, like the not including females or women overall in these studies. They were just like a part of the statistics. But I think that right now there is like a more serious commitment of including females and women in these particular detailed studies. So yeah, the answer is we still don't know. So I think that I hope that if by the end of this session, you have more questions than answers, because that's what drives research, right? We don't know many things. And that's why we're here. We're here to say, yeah, we don't know completely the neurobiology of depression. We don't know completely the neurobiology of depression in women. So that's what makes it very important to study.
Thank you. Any more questions in the audience?
Questions, guys?
Yeah, no?
No.
I'm not sure.
No, it doesn't seem to be that there is question from here. So.
Okay. So, Dr. Aguilar, I have a question.
No question from here.
Okay. It's just that I cannot hear you properly, Dr. Kathy. Okay. Yes or no?
No.
Okay. Because I have another question before the time runs out. We have just a couple of minutes. And this is for Dr. Aguilar, talking about the germline and the germline effect. Do you think that there is a particular phenomenon that contribute to these defects in germlines? Currently, I mean nowadays.
I mean, if there might be like something that is leading to more mutations in the germlines or something like
Yes.
that, or
yes.
Okay.
I'm making reference to more mutations and more defects in meiosis.
Right. Well, that is one possibility. I am not aware of work that has proven in that direction, but we are exposed to probably historically high levels of any contaminants and potentially teratogens in our modern society, despite of going for eating organic and stuff like that. Our environment are considerably more polluted than they were many years ago. So there's certainly the possibility that some of these environmental factors are teratogenic or mutagenic, and that's one possibility. Again, we don't have evidence for, but we don't have evidence against it yet, like strong evidence. So it's something that needs to be looked at. What I would say though, it's like it's interesting that a lot of the mutations, for example, have been concentrated on the X chromosome. That may suggest something perhaps inherently in the X chromosome that makes it more susceptible for mutations. It doesn't necessarily explain everything, obviously, that we know about, for example, autism, but we've known for a while now that there are certain hotspots in the genome where mutations can occur more frequently. And for autism-linked mutations, the X chromosome is one of these hotspots. It's not the only one by any means, but it's one of them. And that also creates this imbalance a little bit, at least contributes to this imbalance in prevalence rates between boys and girls. But yeah, again, another important thing to follow up. What I would say though, because I think this is relevant for the current context, especially, is there is a whole the whole anti-vaccine movement. It gains strength because of these around the world, because of these. There is a false, I think it came from England, that link a certain type of vaccines with the development of autism. The study has been retracted because the author, after several people tried to replicate his findings, they failed. And then eventually the author accepted that he had falsified the data. He had created, basically made up the data. The damage that it created in the public and around the world is huge, because now there is all the anti-vaccine movements are based on that study that is not real. It's a made-up study. It's a fantasy. But all of these, it created all of these, and this is one of these environmental factors that for a while was mentioned. Oh, it's because increase. Since then, there's been so many huge, large, properly controlled studies that have found absolutely no evidence of the link between vaccinations of any type in children and development of autism. But obviously, because we are our brains are wired in a certain way, the general public tends to ignore those findings, or they don't necessarily made it into the general news, so people ignore them. That's an example of something that gone terribly wrong, that arises from badly done research, or actually falsified research, and that has a huge impact in our daily lives. And it's certainly not a factor, if I may say. That's not a thing. It's not real. There's many studies showing there is no link. I'll just say that.
Okay.
There's a question here.
Sorry?
There's a question in the auditorium.
Okay.
Right? Yeah, me over there. Okay, can you hear me?
Yes.
Okay, I have a question regarding the panic attacks. So something that I was reading about is that when we work out, we produce lactic acid, which is something that triggers symptoms in our body, like we heart and all this stuff. But why, if it's supposed that we work out, it's supposed that we can produce neurotransmitters such as serotonin that will help us to relieve this pain of anxiety? Why this lactic acid produces these panic attacks? Oh, internet.
I don't know.
Okay.
I would need to read the studies or the yeah, no idea.
I'm not aware either. Is this something linked to kind of exercise and I don't. Oh yeah, I think I remember a bit about, but I'm not aware of the link very much either. The link between the. I know, for example, if you artificially increase your breathing rate, you can induce certain psychological effects like um, um, like a panic attack kind of thing, or a state of altered consciousness, so to speak, similar to ones some hallucinogenics will have. But I don't know if it's linked to the active lasted metabolism or it's additional changes that happen in your brain. But yeah, there are certain things that you can do to induce altered states of mind, like without necessarily being permanent. I'm not saying like it's permanent, but it's a transient state that you can, for example, that's why breathing, it's modulating your breathing is such an important thing for modulating particularly things like anxiety or acute stress. When you're stressed, the recommendation is breathe slowly. And then if you start breathing fast, you will have, but again, I'm sorry, I don't know either about the connection with lactic acid. That's, you can look it up for next time.
Okay, thank you.
Okay, I think we have time for another question. And I have many, but I will leave just one. Okay, do you guys think that, well, now with the studies that you have done so far, that is there any important gene that can contribute to trigger to other genes to misbehave in terms of the genetic component that these disorders have? So I'm just thinking, for example, like in master transcription regulators, that they enhance or they try to switch on or off other bunch of genes for expressing different proteins. So do you guys think that there are going to be master genes, or at least a couple of genes that can regulate the expression of other genes, specifically in mental disorders?
So in the context of sequencing studies, for example, there are different genes that have been identified, but not at a genetic level, but at the level of gene regulation. So for example, there are many epigenetic factors. For example, one of the genes that like always highlight as important for not only depression, but also like other psychiatric disorders is BDNF, the brain-derived brain-derived neurotrophic factor. I always know the acronym. But yeah, we know that, for example, this is one of the genes that plays a very important role, and there are very regulatory mechanisms that contribute to either being altered or not. I'm trying to think about like the ones that we have described in my lab. There are microRNAs, for example, that regulate expression, but this is more like at a regulatory level, for example.
Dr. Aguilar, do you have something to add? Yeah.
Yeah, sorry, my internet is not very stable. I hope I don't get it cut out. Yeah, the same for autism. There are some transcription factors that are implicated, so they have downstream consequences that sort of like are amplified because of the dysregulation of many genes, additional genes. But also we have a high concentration of genes implicated on translation, not only transcription, but downstream of transcription, the translation from RNA into protein. And again, they impact the level of many gene products, many proteins, throughout chronically. Like you know, if you have a mutation in a negative regulator in several genes, genes implicated in autism are negative regulators of translation. So basically they're breaks, they're slowing down translation, and suddenly you will have an overexpression of a lot of proteins, and their mRNAs might not be changed. Their genes are not changed, but there is an overabundance of those proteins. And that's, for example, a typical something that happens in Fragile X syndrome and tuberous sclerosis. Those are the two key examples, but there are additional factors. So you have impact of certain key regulators in gene expression for sure.
Okay, very good. Well, for finishing this roundtable, I want to say again that we are really grateful to have you both here this afternoon with us. And we hope that this won't be the last time that we see each other. We hope to have you again in our STEM week for next year and the following years. And thank you very much for your time, for your knowledge, and for encouraging our students to pursue research. Maybe not only in Mexico or not only in Latin America, but anywhere in the world. So thank you very much, Dr. Torres Berrio. Thank you very much, Dr. Aguilar Valles, for being here with us. Yes, chapeau to both of you, great researchers. And thank you for accepting our invitation to the STEM Week of Arkansas State University, Querétaro.
Thank you very much for inviting us. Gracias.
Gracias.
Muchas gracias. Que tengan buena tarde.
Thank you very much.
Bye.
Bye. Thank you.